All right, 10:15, right on the dot. So if you just came in, we are recording. Just want to make sure that everybody knows we're recording now. And if you haven't picked up your problem set number 1, they are in alphabetical order. Grades are posted. If for some reason you see a 0, I do have like 3 different homework assignments that there was one that didn't have a full name on there. There was another who just put their student ID on the sheet. And then there was one that didn't have any identification. So if you have a 0, come on up after class, just identify that that's your homework assignment, and then I can go ahead and just enter it in. Okay, you won't lose any points. All right, anybody have any questions about problem set number 1? Okay, so today, remember, problem set number 2 is due today or Thursday? Thursday, all right. Okay, sorry if I scared you. All right, so problem set number 2 is due on Thursday, and that's part 1 and part 2. Okay, everybody have any questions? All right, so like always, let's go ahead and get our top hat devices out. I just have a couple of questions from last week, just to jog your memory. At this point, I want to make sure that everybody feels comfortable with the idea of equilibrium potentials and resting membrane potentials, okay? Last time we talked about the chord conductance equation, so you should now be able to predict the resting membrane potential if you know the relative conductances and the equilibrium potentials. Okay, pretty good so far? All right, so let's start with this one. This is just to, again, see how everyone's doing from last week's material. Go ahead and, there we go. All right, so this one, true or false, at equilibrium, the electrical and chemical work expressions are equal in magnitude but opposite in direction. So on the exam, I've had this question before, and a lot of students have come up to me and asked, what is magnitude? You know, that's just the amount, okay? So you can almost read this question without that term. You can read it, at equilibrium, the electrical and chemical work expressions are equal but opposite in direction. So if that confuses you, just cross it right out. So is this statement true or false? All right, we already have 46 students that have answered. I think we have about 70 in the class, close to. So I will give you all about 10 more seconds. Anyone need a little more time in this room? Having some connection issues? Did you raise your hand back there? No. Oh, okay. All right. All right. So let's go ahead and close this one up. And the correct response is, this is a true statement. Yay, nice job. Everyone got that right. So don't have to review. That tells me that you really know what equilibrium is, that electrochemical equilibrium, and how to think about the equilibrium potential, right? That's the voltage where you are at electrochemical equilibrium, okay? All right, great job. Let's do this one. If the measured membrane voltage that you measure with a voltmeter is equal to the calculated equilibrium potential for potassium, what can you say? A, the net movement of potassium will occur down its concentration gradient. B, there will be no net movement of potassium. C, net movement of potassium will occur against its concentration gradient. And you can talk to your neighbors. If you're maybe a little confused by this, you can definitely talk to your neighbors. That's right. All right, we're up to about 50. So I'm going to give you about 10 more seconds. All right, does anyone in this room need a little more time? All right, so I'm going to close this one, and the correct response is B. All right, most of you got that right. That's wonderful. Let me ask you another question. What do you think the relative conductances would be? Looking at this statement, if the actual membrane voltage is sitting right on top of the equilibrium potential, what would be what could you say about the relative conductance? G. This is a tougher one. You want to turn to your neighbor? Go ahead and talk about it for just a second. Think about the chord conductance equation. I'm looking for a percentage, really. A percentage. All right, does anyone want to take a stab at it? Anyone want to try, educated guess if you aren't sure? Yes? 100%. 100%? Which ion? So, yeah, you're thinking about the right way. So if the actual resting membrane potential is sitting right on top of the equilibrium potential for potassium, yeah. It'd be 100% for potassium. That's right. Okay? So I just want to clarify that, right? The relative conductances would be 100% of the total conductance is due to potassium leaving the cell, right? 100% of the total conductance is due to potassium. That means that potassium is the only ion moving across the membrane, right? This is what Bernstein thought. This was what he thought about the resting membrane potential. And so now you can see that that was actually a pretty good idea, right? He captured the essence of the resting membrane potential. But now we know it's about 90% potassium and 10% sodium. So it pulls the membrane voltage a little bit away from the equilibrium potential for potassium, okay? Any questions? If this isn't clear, then you can come on up after class. We can talk about it a little bit more, maybe draw on the board, okay? But this should be something that you really have grasped, that you have committed to memory at this point, okay? All right, again, if you're still confused, we can always talk about it. With that, you should be able to answer all of the questions now on your problem set number 2, part 1, and part 2, okay? All right, so let's go ahead and move on. Today what we're going to do is we are going to tackle transport. And I understand that this is just a little bit too, far down, but I tried to put this in the back row. Can you see these? Okay, so I'll try to keep it under the screen here. But what we're going to be talking about today is passive, facilitated, and active transport. Okay? So it's a lot of information. I've kind of peppered it with some brain teasers and some top hat questions, some more top hat questions. So don't put that away. So, all right, so let's get started. Okay. We are going to start off with passive diffusion. Now, there's not many slides in passive diffusion. Not too many slides. Here's where we're going to start. Passive transport mechanisms actually include simple diffusion and facilitated diffusion. We're going to start with passive transport, but specifically simple transport, simple diffusion. So in this case, it really doesn't need a protein. These molecules can move across the plasma membrane without a protein. It can, usually these are more hydrophobic molecules. So I will get to that in just a second. Here are your learning objectives. You can take a look at this at home, but I just want to make sure that you know that you have access to it. Okay, so today we're going to actually talk about these 3 different transport properties: passive or simple diffusion, facilitated diffusion, and active transport. And this is a nice slide that actually kind of details some of the major differences. With passive transport, you can actually see there's no protein required. No transporter, no channel, right? It can easily cross the plasma membrane without any issues, right? We'll talk about the flux in just a second and some of the parameters that really play a role in the rate of diffusion with passive transport, but that's where we're going. Facilitated diffusion include ion channels. We're going to get to that in just a second. And permeases, okay? Permeases. Both of those, with facilitated diffusion, a protein is required. These molecules can't cross the plasma membrane very easily. And then finally, if you take a look at this, you can see this is representing our concentration gradient. So in this case, these molecules have a high concentration on the outside relative to the inside. So for passive diffusion and facilitated diffusion, these molecules are going with their concentration gradient or down their concentration gradient, okay? But with active transport, you can see it's the opposite. Active transport protein is required, but you can actually move molecules against their concentration gradient. But again, energy is required. So we're going to be talking about that in just a little bit too. And we're going to focus on the sodium-potassium ATPase, which we've already mentioned before. That's a primary active transporter. So transport mechanisms are distinguished by direction of transport, nature of the carriers, and the role of energy. Okay? So let's get to passive diffusion or simple diffusion. You can actually see here that most of the time when we're talking about passive diffusion, we're talking about small hydrophobic molecules. Small hydrophobic molecules, this includes oxygen. You can see at the top here, this top box, carbon dioxide, nitrogen, some of these gases, benzene. It also includes small uncharged polar molecules, water, glycerol, ethanol. I'm also going to include steroid hormones. Okay, so that's not on this particular slide, but we're talking about hormones like estrogen. These are small hydrophobic molecules. Estrogen can't be sequestered in, say, a vesicle. It can't be, the plasma membrane isn't a barrier to estrogen, okay? So just know that these molecules all can move across the plasma membrane fairly easily. Other molecules, like large uncharged polar molecules or ions, because these are charged, you can see they don't adhere to passive diffusion. Okay, they're repelled here, and that's why they're not included. Proteins are required, so we're going to talk about those when we talk about facilitated diffusion. So let's expand on our knowledge. Now, we have talked about permeability before, right? We talked about it when I talked about the Goldman-Hodgkin-Katz equation. And I told you that there was a real difference between permeability and conductance, okay? A lot of people use these two terms synonymously, but there is a distinction, okay? Like I said, Dave Dawson actually wrote a paper using thiocyanate, and he was able to show that there is a difference between permeability and conductance. Now, just to review, remember, permeability is the ease at which a molecule enters into the pore or into the inner plasma membrane region, which is very hydrophobic. Right? A lot of lipids within that region. So permeability is the ease at which a molecule enters into a pore region or into the plasma membrane, the inner part of the plasma membrane. Conductance is the ease at which a molecule moves across the plasma membrane from the outside all the way into the inside. There is a distinction. And again, you can dissect that out experimentally. Okay, so let's break down permeability, all right? Permeability is equal to the partitioning coefficient. Partition coefficient, that's usually depicted as a beta, okay, times the diffusion coefficient all over delta X, which is the membrane thickness. So you already know something about the diffusion coefficient. Remember that equation KT, that's the thermal agitation of that particular molecule, over 6 pi r is the radius, v is the viscosity. So just so you remember, diffusion coefficient is actually illustrating the solvent and the solute of any solution. That's all incorporated in the diffusion coefficient. And then delta X, you know, is the membrane thickness. Okay, so what is the partition coefficient? How do we think about that? The partition coefficient is basically this. Partition coefficient for any substance is basically the concentration of that substance in oil over the concentration of that substance in water. So here's how to think about it. If that molecule prefers to be in oil, prefers to be in a lipid environment, then this concentration is going to be high. It's going to be higher than 1, right? If that particular molecule prefers to be in water, right? It's basically, if it prefers to be in water, it's hydrophilic. Then basically this equation here will be less than 1, right? So you're really kind of, this equation is illustrating whether a molecule prefers to be in the aqueous solution or prefers to be in a lipid environment, all right? If it's higher than 1, prefers to be in lipid. If it's lower than 1, then it prefers to be in the aqueous solution. Does that make sense to everyone? Okay, perfect. All right, so now it gets a little bit sticky. I'm going to actually go back to Fick's law. So in just a second. And I'll go through this particular diagram down here very systematically to help you through it. All right, so just at the top, this is more descriptive. We're talking about selectivity of non-electrolytes in this case, right? They don't have a charge associated with them. We'll talk about facilitated transport in just a second. So when you are determining the molecular properties responsible for the partition coefficient, usually molecular size of the solute is important. The smaller the size, the more it's able to partition into the membrane. Again, lipid solubility is important, whether this is a hydrophobic molecule or not. And then degree of ionization. Usually molecules that don't have a charge associated with them can enter into that membrane pretty easily. Okay, so there's other factors like temperature and pH that can also influence the extracellular fluid and the ability for a molecule to enter in. But I want you to really understand these 3 properties at the top. All right, so this one's a little bit more difficult. I want you to think back to Fick's law of diffusion. And this is a little bit confusing because they use J instead of F. Remember, I was using net flux, and I was using the F net. In your book, I believe they use J net. Just know that's the same thing. Okay, that's the same thing. So this is net flux, okay? And we're going to start here on your left-hand side, this one right here. All right, so if the beta coefficient is 1, what does that mean? If the beta coefficient is exactly 1, what does that mean? Anybody want to take a guess?
There is no preference.
There's no preference, exactly. The molecule doesn't have any preference, whether it wants to be, right? The molecule doesn't want to do anything. It's teleological thinking, but whether it prefers to be in oil or prefers to be in water, there's no preference. Perfect, okay? So in this case, you can see the concentration outside is greater than the concentration inside. And we're just assuming that the concentration inside the plasma membrane, this is a linear relationship. It doesn't necessarily have to be. Okay, we don't require calculus in this class, so we're not going to go there. But we're just assuming that this is a linear relationship here. Okay, so with this, the concentration gradient, you can actually see outside and inside. Think about Fick's law. The net flux is a certain amount, right? This arrow is, we're going to say it's normalized. And you can see there's just a certain amount of flux due to that concentration gradient. Let's go to the middle panel here. If the beta coefficient is greater than 1, then I already said it prefers to be in a lipid environment, right? It's a hydrophobic molecule. So now let's take a look at what happens to that concentration gradient within the membrane. You can see here's the concentration outside and the concentration inside. Looks the same. However, you can see because these molecules prefer to be right inside the plasma membrane, which is a lipid environment, the concentration bumps up. See where my arrow is? Right in the inner leaflet now, you can see that concentration has bumped up. It's increased because it prefers to be inside that plasma membrane. Now you can see the concentration gradient within the plasma membrane is a lot more steep, right? So if that concentration gradient is steeper, then what happens to the net flux? Think about Fick's law. If the concentration gradient is greater, what happens to net flux? It actually increases quite a bit, right? That's from our problem set number 1. If the concentration gradient increases, then so does the net flux. So that's what's happening here. The net flux increases with those hydrophobic molecules because the concentration gradient within the plasma membrane is steeper. Okay, any questions? All right, so the opposite is true with a hydrophilic molecule or lipophobic, right? So in this case, we're at the very right-hand side here, this one right here. You can actually see that the beta coefficient is less than 1. It prefers to be in the aqueous solution. So you can see here inside the plasma membrane, that concentration has really dropped significantly right in that inner leaflet. Now the concentration gradient is a lot less steep, so it has dramatically decreased. And so now you can see, think about Fick's law, that net flux has also decreased quite a bit. Pretty good? Okay, so now we're bringing in Fick's law to simple diffusion, passive diffusion here. And hopefully this makes sense. So if you have any questions, just come on up after class too. Any questions? Just maybe stop a moment. All right. So here's something that I want you to kind of commit to memory as well. In this case, we're just looking at the green line, right? That's passive or simple diffusion, the green line. So on the y-axis, we're looking at flux. And again, remember that flux is going to be different with different partition coefficients. And on the x-axis is the extracellular solute concentration. So I would say it a different way. I would say this is the concentration gradient, the difference between the concentration outside and the concentration inside, or the delta C, okay? So the greater the delta C, the greater the flux, right? And it's a linear relationship. So in passive diffusion, flux rate is limited only by the concentration gradient. It's a direct relationship. The greater the concentration gradient, the greater the flux. Okay? Not too bad, right? So let's just start building our chart here. What are some of the parameters involved with passive transport? Building our chart, just review. Protein required? No. No protein. What else?
It's based on concentration gradient?
Yep, based on the concentration gradient is important. So the concentration gradient is basically determining the net flux. Okay. Some of the molecules that participate in passive diffusion: hydrophobic molecules, usually small, non-electrolytes, pretty good. All right. So, is energy needed? No energy. No energy is needed. All right. So it depends on the concentration gradient. How is the net flux going to proceed? Low to high, high to low? Is it going to go with its concentration gradient or against? It'll go with its concentration gradient. From high to low. All right. So we'll get to saturation in a minute. I'll add that to the chart. But in this case, no energy is needed. Depends on the concentration gradient. It's always going to go from high to low. The steeper the concentration gradient, the higher the rate of diffusion, the higher the net flux. And in this case, saturation does not occur. But don't really know what saturation is yet, but we'll get to that in just a second. All right, so let's just move on to facilitated diffusion. Here we're going to talk about ion channels, one of my favorite topics. Love ion channels. All right, let's see, here we go. All right, so again, here is our roadmap. This is what we're going to be covering right now. Ion channels, we'll talk a little bit about voltage-gated sodium and potassium channels. And then I'll also talk about CFTR, that's the cystic fibrosis transmembrane conductance regulator. You have no idea what I'm talking about right now. But it is the channel, when it's not inserted into the membrane, it is responsible for the disease cystic fibrosis. Okay, that's our next problem set, actually. We'll be talking about cystic fibrosis. And then carriers. And again, if you want to take a look at those learning objectives, now we're on those second ones. Okay, so facilitated transport. Basically, channel is required. A lot of times these are molecules that do have a charge associated with them. okay? And in this case, still no energy is required, just a protein. Again, it depends on the concentration gradient. It's always going to go from high to low. But in this case, we'll be talking about saturation in just a second. Saturation is possible. All right, so let's take a look at what exactly is saturation. So with simple or passive diffusion, we were illustrating that with the green line here. With saturation, basically this is where there is a maximal flux. And you can see with this graph, we're talking about the purple line. This is facilitated diffusion, okay? So, what do you think when you think about a protein and molecules either going through an ion channel or a carrier where there's a turnover? Why do you think saturation occurs? Why is there this plateau, this saturation? What could be limiting diffusion? Yeah?
Because there's a set number of proteins that are going to be going across the membrane versus just diffusing.
That's right.
Molecules.
Right. So basically, you're limited by the number of proteins that are inserted into the membrane, right? And what else? Kind of going along the same idea. You're talking about carriers. So it's the number of channels and carriers, and what else? You guys know Vmax, like the turnover rate, right? You're also limited by that turnover rate. Okay, so those are the two things that causes that plateau, that saturation. So saturation does occur. In carrier-mediated transporters, especially, the number of available carriers places an upper limit on the flux. I would also add to this figure the turnover rate of each carrier. Okay, perfect. So let's take a look at this. With this one, don't worry about your top hat questions. This was a top hat question, but we don't have to worry about getting it out. Do you think ion channels are saturable? Basically, they allow for 1,000 times more molecules to move across the membrane than a carrier. Okay, so do you think they're saturable? It's kind of a tricky question. Your book says no, but experimentally, this is not physiologically relevant. You can actually create such a concentration gradient that you can saturate certain ion channels, especially the calcium channels, where calcium enters into the pore in single file. So if you have a steep enough concentration gradient, yeah, you can actually saturate out an ion channel. Okay, it's a tricky question because your book does say no. All right, so let's move on to ion channels, okay? Let's talk about some of the properties. Proteins that span the membrane allow for the flux of about 10 million ions per second through a pore is an ion channel. That's an enormous amount of electrolytes. Yes?
So then what does the concentration gradient have to be for it to saturate?
Oh, I don't know that off the top of my head. I can look that up. Yeah, I know specifically, though, calcium channels are unique. And like I said, they go through the pore in the single file. Right. All right. And there's multiple calcium in each channel. Not like some of the channels we're going to be talking about in just a second. All right, so ion channels usually transport more ions per second. You can see that 10 million in contrast to the pump, which only is 1,000 ions per second. So carries a lot more molecules across the membrane. Now, what's really important and why we're going into detail about ion channels is that it's very important in excitability of cells. And when we're talking about excitable cells, we're going to be talking about neurons. And then for exam 2, we're going to be talking about muscle. So a lot of people may not realize that muscle are excitable cells as well. They actually have action potentials. Okay? So they regulate nervous tissue, muscle contraction, hormone secretion, and other important roles. All right, so there's 3 things that I want you to kind of put to memory about ion channels. 3 things. First is channels assume different conformational states. We're going to, this is titled Conductance. Okay, so again, conductance is the ease at which an ion moves across the membrane. And it has everything to do with the whether this channel can conduct ions or not. They can assume different conformational states. If they're open, they're conducting. If they're inactivated or closed, they're non-conducting. Okay? Then we're going to talk about gating. So with this next one here, channels open in response to a specific stimuli. This is called gating. All right, so ion channels have gating properties. Voltage-gated ion channels, they actually open in response to a change in the membrane voltage. Okay? Mechanical stimuli can also open channels. So imagine if I had the ability, we had a big cell right in front of me, and I could take a pencil or a pipette and make a little dimple on that cell, like mechanically push into it. I could open mechanically stimulated ion channels. So they can open that way. Mechanical stimuli. Phosphorylation, a lot of times ion channels open if they're phosphorylated. What that means is a phosphate group is attached to the protein. There's a conformational change, and it opens the channel. Phosphorylation. And then we're also going to be talking about ligand-gated receptors, which are ion channels. Like neurotransmitters can bind to its receptor, and it actually is an ion channel. It opens up a pore region and allows for molecules to move through. So those are ligand-gated. All right, and then most ion channels exhibit selectivity. So what that means is they can allow for sodium to move through, but they don't allow potassium to move out. Right? So they're very selective for one ion, but not another. And we're going to talk more about that in just a second, too. So I've shown this over the years. Can you see this? No, you can't see this yet. Hold on. Let me get out of this. LOL. When I saw this video many years ago, it's so silly. It's just funny. So students were told to make a video about selectivity of an ion, and this is what they came up with, and I just laughed. I just thought this was funny.
Membrane. I let some things in, and don't let other things in. For example. No, you cannot go ahead. You, go ahead. No. There. See how I let Laura in, and I did not let him in.
I like that the molecule fights back. And the one who goes through actually turns around and goes, ha ha. Anyway, I just thought that was really funny. I better give credit to where credit is due too. So anyway, that's selective permeability. Basically the ability for...
Okay, so in this video, we're going to go over some of the common organelles of a eukaryotic cell. Now, an organelle is simply a word that means...
Okay, so basically, when we're talking about selectivity, you're allowing for one molecule to move across the membrane, but you're not allowing for a different type of molecule to move across the membrane. So highly selective ion channels, like voltage-gated sodium channels, we're going to learn a little bit about their biophysical properties. All right, so going back to our, it's a little clunky here. Okay. So, That's called selectivity. So the three different properties that I want you to know for ion channels: conductance, gating, and selectivity. All right, so let's start with conductance first. Again, channels have the capability to be either in a conducting state if they're open, or a non-conducting state. And there's multiple different states that basically cause the channel to not allow for molecules to move across the membrane, non-conducting states. All right? Any questions about conducting and non-conducting? Hopefully you all know conductance and what that means at this point. We'll revisit this, and when we're going to specifically talk about voltage-gated sodium channels. All right, so let's talk about the different different gating properties now. Move right to gating. We're going to talk about 4 again: voltage-gated ion channels. So the voltage-gated ion channels, let me blow this up a little bit. Talked a little bit about this already. All right, so voltage-gated ion channels actually open in response to a change in membrane voltage. So for voltage-gated sodium channels, remember at the rest, the inside of the cell is usually negative, outside positive, okay? And if you impose a change in the membrane voltage, now the inside is positive. You've basically depolarized the cell. It's a lot more positive inside. You can see that causes the channel to open. There's a conformational change, and it causes the channel to open. We'll talk more about voltage-gated sodium channels in just a second, but basically there is this region within the channel that has all of these positive residues. So when there's a change in the membrane voltage, it causes the S4 region to twist up and open the channel. Okay? So it kind of repels that positive charge inside the cell. It twists up and it opens the channel. Okay? So these are voltage-gated channels. All right, mechanically gated channels are also called stretch-gated or tension-gated. They have these elements on the channel that are attached to cytoskeletal elements. So if I took a pipette and pushed down on the cell, it stretches it and opens it. So what other scenario could open up a mechanically gated channel? What if you dropped a cell in a hypotonic solution? What's going to happen? It's going to swell, and it's going to open up these channels. Right? We're actually going to talk about cell volume regulation next week. Okay? So, if the cell starts to swell, it's also going to stretch and open up these mechanically gated channels, these stretch or tension gated. These channels are going to desperately move solute out, trying to move water out at the same time to keep its volume within a homeostatic range so it doesn't burst. A lot of times that doesn't work. Cells lyse all the time, but that's what's happening. They're trying to regulate their volume. All right, phosphorylation gated. I'll just add to this one. CFTR is a channel that's phosphorylation gated. So we'll talk more about that, but you can see here, once a phosphate group is added, it opens up the channel. And then ligand-gated channels. The ligand attaches to the channel, and it opens up in response. There's a conformational change. So those are the 4 types of gating that I'd like you to know. And now I'm going to focus on the voltage-gated sodium channel. We're going to go back, revisit this voltage-gated channel. And the reason why I'm going into detail about this channel is because it's going to be really important when we talk about action potentials. So don't purge any of this. Okay, so specifically looking at voltage-gated sodium channels, basically this has 3 different states. C is the closed state. O is an open state, and I is called an inactivated state. So going back to conductance, the closed or the inactivated state are both non-conducting. Only the O, the open state, is conducting. Is a conducting state. Okay, so now let's take a look at the process of activation. Activation is just a term that we use when the channel goes from a closed state to an open state. It's a transition to a conducting state. That's called activation. If we go from the open state to the inactivated state, transition to a non-conducting state, this is called inactivation. Okay? And then from I to C is recovery from inactivation. Okay, so that's a transition to the closed state. Now you're probably wondering, why do we have both non-conducting state, an inactivated state, and a closed state? So the reason why is we're going to be talking about refractory period when we get to an action potential. If the cell is in an inactivated state, no matter how strong a second stimulus is, it's inactivated. It can't open. It's like frozen. No matter how strong a second stimulus is, you can't open the channel. You actually have to recover from inactivation to the closed state in order to open the channel. You have to recover from inactivation. So let's take a look at another animation. Okay. All right. You can't see this. People at home can see it, actually. Let's get out of this. I think this will help. It'll let you see in real time, like a cartoon, how to think about activation, inactivation, and recovery. All right, so here's our voltage-gated sodium channel. It's basically the brown one on the side there. Okay, it's on a loop, so we'll come back to it. This right here in the middle is the activation gate, and this gate right here is the inactivation gate. Okay, so let's see what happens. We'll get to the closed state. The inactivation gate is open. This is right here, the activation gate. Stimulus opens the gate, now it's conducting, then it inactivates. And then it has to recover from inactivation before it can open again. Do you want to see that cartoon one more time? Does that make sense? So this is the activation gate right here, and this is the inactivation gate. All right, so we'll start again. It's closed. Now it's activated, that's activation, and then inactivates. This inactivates. And then it has to recover from inactivation before it can open again. Confused or pretty clear? Pretty clear? Okay, I see some heads nodding up and down. Those are the three different states of the voltage-gated sodium channel. So you need to think about that, especially when we talk about refractory periods, okay? So again, we're just learning things in a very compartmentalized way, but we'll put it all together at the end, right before exam one. All right, so this is some text just to help you when you get home. Learn about activation, inactivation, and recovery from inactivation. Now, just so you know, experimentally, you can drive the cell from the open to the closed state, but it doesn't happen normally. It's not physiologically relevant, but in the laboratory, you can impose such a negative voltage that it won't go to the inactivated state. It'll be driven straight from the open state to the closed state. This is called deactivation, all right? And that is written right here. It talks about deactivation as well. All right, so when you get home, you can read this, and then hopefully that becomes a little bit more clear. All right, So let's talk about selectivity now. We actually talked about conductance, gating, and now selectivity. What determines selectivity? There's a few things. Size. The larger the molecule, the harder it is to get into ion channels that only allow for small electrolytes to go through. So size can prevent certain molecules from entering into the pore. Interaction of charges within the pore. So take a look at this. You can see that there are amino acids, residues, that line the inside of the pore region of an ion channel. They are all positive in nature. Okay, so what it's going to do is it's going to attract anions and repel cations. So this is a typical anion channel. Probably conducts chloride, bicarbonate ions. Okay, so you can see the interaction of charges within the pore are going to cause this particular channel to become more selective. It's going to allow for anions, but repel cations. So ease of dehydration. This is probably a new one. All right? When you drop an ion, say, into a solution, you may or may not have known that water molecules are polar molecules, right? They actually surround each and every ion. And before that ion moves through this pore, it has to rip off the water molecules first. That's the ease of dehydration. So let's get out your top hat questions here. Oh, I think I gave you the answer already. Maybe you didn't see it. Let's see here. All right. Oh, I don't have it on top hat. Let's just answer the question without worrying about top hat. Sorry about that. Okay. All right, so think about this. Here's the scenario. Think about an ion channel that allows for potassium to move across the membrane, but not sodium. Okay? So in this case, which ion? Both of them have a positive charge, right? Here we go. Which ion is smaller? Which ion is smaller? Do you know? Think back to chemistry. What is it? Sodium. All right, sodium is a smaller ion. That's correct. Sodium actually has a radius of 0.95. Potassium has a radius of 1.33 angstroms. So sodium is a smaller ion. Think about an ion channel. How is it possible for it to repel sodium, which is smaller? Both of them have a positive charge. How is it possible that you can have a highly selective potassium channel? Sodium smaller, both of them have a positive charge. How is it possible? Right? It's kind of a brain teaser in some ways, right? The only other characteristic we were talking about with selectivity was the ease of dehydration, right? It's actually easier to rip off the water molecules that are surrounding a potassium channel than it is a sodium channel. In other words, the sodium ions are hanging on to the water tighter than the potassium is. Okay, so it's easier. Take a look at this. I think this is actually really interesting. When you look at the inner portion of an ion channel, the pore region, you can see that there's these carbonyl groups here. They have oxygen attached to it. And it is exactly mimicking the potassium ion surrounded by water, which is right here at the bottom here. Each one of these are representing a water molecule, right? H2O, H2O, right? So you actually have, with the potassium ion, these water molecules they look something like this in this orientation. What happens with potassium, as it slides into the channel, the pore region is actually exactly mimicking the water molecules. So it slides directly from the water molecules right into the pore region, right? It's actually really, really elegant. The sodium channel is too small. You would actually have to inject energy in order to close that pore region just slightly in order for it to be more selective for sodium. So sodium actually hangs onto those water molecules tighter, can't rip them off, and it doesn't enter into that highly selective potassium channel. All right? So, very interesting. The fixed filter structure is finely tuned to accommodate potassium, and for sodium, it can't shrink enough to properly bind the smaller sodium ions. It can't rip that water off too. So the energetic cost for dehydration is higher for sodium. Again, it's all about that ease of dehydration. Okay? So that's selectivity is achieved. When I think about this is really something else. To me, this is very elegant, that it is able to be highly selective for potassium and not sodium. So this is just a cartoon, really. I mean, just a funny. Humans found a similar solution to a similar problem. How to pass big feet blocking small feet. Have you ever seen cattle grids in Europe? Right? With the cattle grids, you actually have, the cattle have very small hooves, which would symbolize the sodium. Humans have lot bigger feet, and that would represent potassium. With these cattle grids, humans can walk across it, or cars, pretty easily, but it keeps out the cattle. See, they won't actually step on it because their feet would actually go through. So, just another way to think about selectivity. There's some beautiful cattle grids in Europe. Yeah?
What do they do if the cattle decide that they want to go across it?
I think I hope they haven't learned the hard way. But I guess I've imagined that maybe their foot has slipped in and that they really know what's happening so that they don't enter into it. But basically, that's what we're talking about here. So, I just think this is cute. I don't know how to think about this. That's a little puppy that's just jumping over the cattle grid. So, are there any questions about conductance, gating, selectivity of ion channels? These are kind of the foundation, fundamentals of ion channels, which we will use when we get to action potentials. All right, questions? Better stop. Okay. All right, so some formal terms again that I want to throw out, a little bit of brain teaser. I've picked this one up from different textbooks too. This was, I think, from a Pearson textbook. I want to introduce the idea of depolarization and hyperpolarization. Another terminology is big. So, in this case, if you open up a sodium channel, and if the concentration gradient and, remember, the electrical force are usually like at the resting membrane potential, then sodium will enter into the cell, right? Sodium's going to enter into the cell. And what it's going to do is it's going to pull the membrane voltage closer to the equilibrium potential for sodium. When it moves in a positive direction, it is called depolarization. Okay? Now I'm going to start using the term depolarization instead of moving in a positive direction. If we open a potassium channel, potassium leaves the cell, that positive charge is leaving the cell. That's going to pull the membrane voltage closer to the equilibrium potential for potassium. When it moves in a negative direction, this is called hyperpolarization. Okay? You can also use this term like relative. If you say this cell is more hyperpolarized than another cell, then you know that this one has a more negative potential than the other. Okay? All right. So, what about this middle panel? What's wrong with this middle panel? What's wrong with this middle panel? Yes. That's right. There's no molecules moving across the membrane. There's no ions, there's no channels, there's no ions moving across the membrane. So what should the membrane voltage be? You guys remember the computer simulation? If there's no ions, you can even think about chord conductance equation or even the Goldman-Hodgkin caps. If there's no ions moving across, it'd just be zero, right? If there's no ions moving across the membrane, it should be zero. So this is not correct. That's why I kind of, just be very careful. What I have learned is some textbooks, or if you're looking at websites, animations, a lot of times there's misinformation when it comes to physiology. You have a more sophisticated view of physiology now, so I do expect you to catch some of these mistakes, okay? All right, so this is the last slide for ion channels. This is a crazy slide, but this is giving you like the voltage-gated ion channel family, all right? There's more than 140 members. Their conductance varies by 100-fold, you know. These right here, I'm going to start right here, the K2P channels. These are the leak potassium channels that set the resting membrane potential. Okay, so I'm putting a face to a name. All right, that is just a leak channel. It's always open, setting the resting membrane potential. Here's our voltage-gated potassium channels. This one we're going to actually talk about when we get to the heart. These are hyperpolarization cyclic nucleotide-gated channels. These are your pacemaker cells. These are driving your heartbeat. They're called Hcm channels. Actually, what's funny about it is they are called funny channels because they were very strange when characterized. So if you look at a journal article, when it says funny channels, that's what these are. The Hcm channels are called funny channels. Not kidding. Funny, strange, not funny, haha. Okay, here's the sodium channel and calcium channels. They're very large channels. And I just wanted to kind of talk about these two right here on the side. These are called trip channels, which I think are fascinating. The trip V channel right here, vanilloid trip V. These are in your mouth. They're actually temperature sensitive. So they will open if you drink like hot tea or something like that. It kind of tells your mouth that there's some hot substance in your mouth, okay? But if you ever tried chili peppers, why does it feel hot in your mouth when you eat chili peppers? It's because they're activating these trip vanilloid capsaicin receptors. Does the same thing. These trip M channels, I think, are fascinating too, right up here, trip M. These detect cold temperature. Have you ever wondered why when you eat a menthol it feels cold in your mouth? It's because it's activating these TRPM receptors, the same temperature sensitive receptors. Okay, pretty cool, right? All right, so these are part of the voltage gated ion channel superfamily. Also here, the permeases. Let's just finish this up with the permease. We've concluded the ion channels. Permeases actually have a turnover rate. A perfect example of this is a glucose transporter. Glucose binds to the transporter, conformational change, and then it delivers the glucose inside the cell. SGLT1, we'll talk about that when we get to the gut. Okay, so it's actually transporting glucose out of the small intestines into eventually the bloodstream, right? Elevating your blood glucose levels. All right, so again, there's a conformational change that occurs. Solute acts as a ligand, binds to the transporter, and then a subsequent shape change in the protein releases it on the other side. Okay, this is where we will use our top hat devices. And I'm not going to open this up completely. Get to active transport. We're going to be talking about the difference between primary and secondary active transport. Okay, so here we are here. Active transport, primary and secondary. All right. So I'll open this up pretty wide. Here we are here. Remember, with active transport, energy is required. Actually, I think we're running out of time, but you can fill this out at home too. I do want you to know the differences. With active transport, energy is required, but you can move solute from low concentration to high concentration. You can actually move molecules against their concentration gradient. Pumps do that. They use ATP. Or sometimes you have these exchangers where it's using the gradient from one molecule, like the sodium gradient, to move a different molecule against its concentration gradient, right? You're using that energy of the sodium gradient. So at the bottom here, solutes move opposite to the direction predicted by the concentration gradient. Active transport, protein transporter is needed, energy is required, molecules can move from low to high. So that's how you would fill out the rest of that chart. The difference between primary active transport and secondary active transport is that primary active transport uses the reaction that releases energy, right? It actually uses, say, the hydrolysis of ATP. When ATP is broken down into ADP and pi, that energy that's released is harnessed to move molecules against their concentration gradient. Exergonic reaction. Secondary active transport couples the movement of one molecule to the movement of another molecule. So it uses a lot of times the sodium gradient. Sodium moves down its concentration gradient, and then it moves another molecule against its concentration gradient. So let's concentrate with primary active transporter first. Okay, there's 3 types. P means phosphorylation type. Classic example of P type is the pump, the sodium potassium ATPase. F and V type. F means factor type and vesicle type. V is for vesicle, and a lot of times there's compartments within the cell that pump hydrogen into vesicular regions, lysosomes, and it makes a pH change within that vesicle. Most of the time they're pumping protons, hydrogen ions. And then ABC type. ABC means ATP binding cassette. It actually binds ATP. A lot of times these carry large organic molecules. CFTR is actually a type of ABC molecule. All right, so we've already talked about P type. We're going to concentrate on the pump for a minute. All right, so here's how it works. This is 411 in your textbook. Can you see that all right, or should I blow this up a bit? Blow it up a bit? Nope. Try this down here. Is that better? Okay, so remember, with the pump, just put this right here, with the pump, for every turn of the pump, you're basically removing 3 sodium ions, transporting it out of the cell, and you're bringing in 2 potassium. This is called electrogenic, because for every turn of the pump, you're removing 1 positive ion, right? The net movement is 1 positive ion out of the cell, okay? So 3 sodium basically attach, you can see, from the intracellular side. This is a little backwards from some of the other diagrams. So the top here is inside the cell, and the bottom here is outside the cell. So sodium binds to the pump. You can see here ATP attaches. There is a conformational change once ATP is hydrolyzed. Okay, so You can see here ATP is hydrolyzed to a phosphate group, an ADP. There's a conformational change, and sodium is delivered to the extracellular compartment outside the cell. Okay? Now, there's a low affinity for sodium and a high affinity for potassium. Potassium binds to the pump, and then once ATP, that phosphate group, is released and ATP binds, there is a conformational change that delivers to potassium to the inside of the cell. Okay? So we have a few minutes here. Oh, on the exam, think about this. I try to give you a heads up on what you can expect. Okay? If you see something that looks like this, I do want you to be able to label it. All right? So 3 sodium out, 2 potassium in. ATP is hydrolyzed to ADP. A phosphate group remains on the pump, and then the phosphate group is released and ATP again binds. Okay? Okay. So here's our brain teasers. I want you to think about this. Here is an interesting way to think about the pump. If we block the pump with a cardiac glycoside like wobane, this is just an inhibitor of the pump, what do you think will happen to membrane voltage? Just have a few minutes here. You can just turn to your neighbor. I think it'll take too long to do the top hat. Remember what we're talking about here. This is electrogenic. So what's going to happen is A, the membrane voltage is going to become more positive inside. It'll depolarize. Or will membrane voltage stay the same? Or will it hyperpolarize, become more negative? So what we're talking about here is with every turn of the pump, something happens, and we're inhibiting this. What's going to happen to membrane voltage if we add this drug? You guys want to take a guess at it? What do you think? A, right? Because what's going to happen is you're going to start to accumulate positive charge inside the cell, right? And that's exactly what happens in the lab. If you actually add wobane to cells, it causes depolarization. All right? Let's take a look at how are we doing on time. We got a couple more minutes. All right. Let's take a look at this one. If we block with the same drug, what happens to cell volume? Ooh, what happens to cell volume?
Wait, so positive charge inside the cell, one would flow outside the cell, inside the cell.
So think about this in terms of osmolytes now. Osmolytes, osmotic pressure, osmolytes. With every turn of the pump, you're actually removing one osmolyte. If you block it, osmolytes are going to accumulate. Water's going to move into the cell. It's actually going to swell. All right? All right. I don't think we have time for this last one. This is what we'll pick up. In fact, this will be our top hat question for Thursday. All right? All right, everybody. I think we're going to end it there. It's 11:29. Have a good Wednesday. I'll see you on Thursday.
I have a question about driving force.
Okay.
Like, what is the equation for that?
Okay. It's actually pretty easy.
Okay.
So if you take a look at like the equilibrium potential for sodium and the resting membrane potential is down here, and the equilibrium potential for potassium. So Vm minus Es is the driving force. So it would be, in this case, this is minus 60 and this is plus 61. For sodium, it's going to be minus 70 minus 61. It's minus 70, minus 61. That is okay. Yeah. Right? Because it's going to be minus 70, minus 61, and it's going to be minus 131. It's going to be huge. So what you can say is this is the driving force. Right? This is a very large driving force. So for potassium, it's actually going to be minus 70 minus, let's just say it's,
Oh, I'm sure they did.
this is going to be plus minus 70 plus 80. So that's going to be 10. So it's small. Yeah. You're welcome. Yes. Hi.
Hey, can you explain the extra top-out question that we did at the beginning?
Yeah.
Where it's right on
Yes.
the resting membrane potential equal to this one? Yeah. Okay, so let's just say it's a 2 ion. Just look at
Relative conductance for potassium. Plus conductance for if this is
Recently drop her off
100%,
I would have been
I was
outside.
driving. Did you drive here?
Yeah.
right? Then this actually just cancels out. Because this was just one.
God, I just need some
Then
sleep.
the equilibrium potential for potassium is 2.
She was like, I was supposed to be up at 8:30. I was like,
But when this the rest of the large
she was like, yeah, I missed my quiz. Can you drive me to my class? I was like,
There is no net
it was like 9:30.
of everything
She was like,
but
before the class.
potassium or
I don't know.
potassium?
I was so confused. I was like, just don't go.
Like potassium's the only
She was like, drive me.
Yes.
Or there's no net movement of potassium.
I'm gonna be late to my class.
Okay.
Does that make sense?
And then I
Yes.
couldn't find a parking
So
spot.
only potassium is moving across the membrane. Okay. Only potassium. And if that's the case, then the electrical force and the chemical force are going to be exactly the same. There's going to be no net movement. So you're absolutely right. Potassium is moving, but it's the only ion, and it already has reached electrochemical equilibrium, so there's no net. Does that make sense? Okay, so any time the resting membrane potential is actually sitting right on top of the equilibrium potential, you can assume that 100% of the conductance is due to that exact okay. So if the resting membrane potential was sitting right here on the equilibrium potential for sodium, you can imagine that there's nothing else but sodium moving into the cell. Okay, and it is at its electrochemical equilibrium. There's no net. Okay, but anywhere else, you know there's net movement, but you're at a steady state. So here, basically what you're looking at is that 90% of the conductance is due to potassium, only 10% is due to sodium, and you're at steady state, so that the current for sodium is equal to the current for potassium in the opposite direction. That's a steady state.
So if this was say closer to here, there'd be more
Yeah.
of the sodium moving back.
That's right. Okay. So you can assume if the resting membrane potential is here, even before you run that to these numbers, you can say, oh, I can see that about 90% is due to sodium and only 10%. Okay. That makes sense. Thank you. Good. You're welcome.
Hi. So
Hi.
I won't be here Thursday.
Okay, sure. Yeah, that's fine. Yes, thank you. Questions?
Yeah. So we couldn't find the project thing online.
I will get that in there. It's not entered yet.
Okay.
So I'll work with Magna to do that.
Okay.
Yes.
Awesome. And then I'm not gonna be here Thursday
Okay.
because we're going for livestock judging in
Sure.
Mississippi.
Sure.
Can I just turn in my homework on Tuesday? Is that fine, or
Yeah, or you can email it, or you can, we
just
can
email it to you,
or Magna.
Okay.
Yeah, email it to Magna. I think that's going to be better.
Okay,
And then she'll
awesome.
just put it into the.
Awesome.
Yeah.
Okay. Thank you.
No worries.
I have one of the problems that doesn't have a
Oh, okay, sure. Yeah, take a look at this. Is that one yours? That one yours?
This one.
Okay, no
Way to
worries.
write my name on it,
Yes.
Sorry.
Hi.
Oh, thanks.
I just have this group.
Okay,
we were
yeah.
the group that wasn't here.
That's
we
right.
also didn't come up with a name yet.
Thank you for doing that.
Yeah,
I
can we
really
just
appreciate
email you a name because we didn't
Yeah,
decide on it
yeah,
yet.
no worries.
Okay.
Yeah, thanks for turning that in. I appreciate it.
I have a question about the part 2.
Okay, sure.
I've heard the questions down. When I asked to calculate the magnitudes, and I wasn't sure if I was supposed to use the Golden equation or something else.
I would use this one right here. This is the chord conductance equation. And so it's going to be Vm, that's the membrane voltage, is equal to the relative conductance. So how many channels are open? Usually at rest, 90% of the total conductance is due to potassium leaving the cell. 90%. So you put 0.9 and then multiply that by the equilibrium potential. And then for this one, it'd be 10%, which would be 0.1, and multiply that by the equilibrium potential. So I would use the chord conductance equation. That's a really good way to predict actual membrane voltage.
And then for this one, I wanted to get the calcium, and I wasn't sure if I should use this one.
Yes, so you got that right too. You got the valence is plus 2. And then yeah, you would use the same thing. Okay, yep. Okay. Does that one not have the actual resting membrane potential listed in the question now?
I don't think I
Okay.
calculated.
Okay. Yeah, if there's already a stated membrane potential, use that. But yeah, I can't remember.
I think because it was because I wrote down.
Okay.
Figure it out. Yeah.
Okay. Okay.
And then I also want to ask you about the scuba.
Oh yeah. I'm
So-
gonna get us together as soon as I get that final list together,
Okay.
and we'll have a preliminary meeting, and we'll talk about everything you need to do.
Okay, because I
If
have
you-
a neighbor who's a scuba instructor.
Perfect.
And she's like, you probably should sign up as soon as possible, but I
Okay.
don't know.
Okay.
That's what I wanted.
I would say
don't know.
then, if you have a friend, if she can give you some direction on it, I always tell students to go to Midwest Diving. Okay. Mark is the one that owns that, and he gives students discounts. So if you want to do that too, if you can get in sometime in April, right before we leave, you can do your book work and pool work basically in 1 weekend, 1 or 2 weekends, and then you can just get down there. Does that sound okay?
Yeah, sounds good.
So I'll send out an email soon. I'm just waiting for that final list.
Okay.
Okay. Yeah. That's
You know
awesome.
How many is there will be?
Divers? Don't know yet.
Well,
Oh,
as
there's 24 of us.
Oh, that's
Yes, yeah. I don't know the number, the ratio of divers to
Oh, the divers,
snorkelers
that's
yet.
okay. I just wanted
Okay.
to know how many
Yeah.
people were going.
Yes, yeah.
Sounds good.
Yeah. Hi, Maria. Sorry about yesterday. So I do record every lecture, in fact. Let me go ahead and stop this one. Is it okay?